Kindly bear with us. There will be a delay of another 3 to 5 minutes because we are waiting for the PhD students of IRA Mega University Hub who are doing their PhD in entomology to join. The master students have already joined us and they are here with us in the committee room. So kindly bear with us. We'll start the talk maybe in another 3 to 5 minutes. Okay. Thank you. Dear colleagues from IIRR, colleagues from Indian Institute of Millet Research, Indian Institute of Oilseeds Research, and my dear colleagues who are members of Society for Advancement of Rice Research, and also very valued or esteemed students of MSc in entomology and also PhD in entomology. I welcome each one of you for a short talk that will be delivered by Dr. Ravi Kiran Donthu, who's serving as assistant professor in computational genomics, Center for Life Sciences, Mahindra University, Hyderabad, India. The reason why it should be of interest to both entomologists and also geneticists and also to biotechnologists is the topic. He'll be speaking on adaptive evolution in agricultural pests and honeybee diversity, a genomics perspective. And I think understand that he has done pioneering work on this particular topic in his postdoctoral studies. And Dr. Ravi Kiran will be talking on this particular topic. But before we move ahead further, I request my colleague, Dr. Chitra Shankar, who is principal scientist, entomology, and also serving very actively as a faculty member of IRA Mega University in entomology subject, to kindly introduce the speaker to all of us.
Thank you, sir. It is my honor to introduce the speaker of today. Dr. Ravi Kiran is an assistant professor in computational genomics, and is presently working at the Center for Life Sciences, Mahindra University, Hyderabad. And he has 20 years of advanced research, analysis, and teaching experience in genomics and bioinformatics. Prior to this, he was working as senior associate professor at Atlas Kill University, Hyderabad, India. He completed his Bachelor of Science in Pharmacy from JNTU, Hyderabad, and did his Master of Science in Bioinformatics from the University of Leeds, United Kingdom, on the thesis topic, Web Interface of Protein Ligand Interactions and 3D Visualization Tools. And he did his PhD in Cattle Genomics and Bioinformatics from the University of Illinois, Urbana-Champaign. And his title during his doctoral was Fine Mapping and Evolution of a QTL Region on a Cattle Chromosome 3. Earlier, for graduate and undergraduate students at Florida International University, Miami, Florida, and the University of Puerto Rico, San Juan, Puerto Rico. He has worked on various interesting grant fund projects on honeybee genetics and health diagnostic panels, impact of unpredictable population-wide disturbances on evolutionary processes in honeybees in Puerto Rico after Hurricane Maria, and assessment and preservation of disease-resistant honeybees in Puerto Rico, certification of Puerto Rico honeybees, and also various other projects. He has numerous peer-reviewed publications in high-impact journals, including Current Opinion in Insect Science, Insect Biochemistry and Molecular Biology, PLOS Computational Biology, Physiological Genomics, etc., and on some excellent titillating topics such as genome scan detections of selective sweep among biotypes of soil with Aphis glycinus with differing virulence to resistance to Aphis glycinus in soybean cultivars. Direct sequencing of insect symbionts via nanopore adaptive sampling, and description of soybean aphid, Aphis glycine, mitochondrial genome, and comparative metagenomics of aphididae. We are very happy that he was ready to give a talk about his lead to future collaborations. To Kiran now. Thank you.
Thank you, Dr. Chitrama, for the wonderful introduction. Yeah, thank you all for coming, for taking time to join for this presentation. We can record. Yeah, I would like to thank you everyone for taking time to join this talk. Today I am going to talk about the research work that I have done in the last 5 to 6 years in insect genomics. So prior to coming to, I joined Mahindra University recently, like in July. Before that, I was like in the last 20 years, I've been at University of Illinois, Urbana-Champaign, where I did my PhD, and then I started working there. And like till 2017, I was working there, but then remotely I was working for University of Puerto Rico and Florida International University, living in Illinois. So as Dr. Chitra ma'am already described, I did my bachelor's in pharmacy from Hyderabad, and then I did my master's in bioinformatics in 2003, and then I moved to US to do my PhD in genomics and bioinformatics area. So, like after I graduated from University of Illinois, in the same university, I joined in the Biotechnology Center, where we get to work with PIs from different departments and other campuses in US and other places. And so from 2009 to 2017, I was like a bioinformatics research scientist. We're working full-time on various genomic and bioinformatics research projects, primarily from next generation sequencing data to different types of analysis, like including genome sequence assemblies, transcriptome assemblies, RNA-seq data analysis, metagenomic analysis, and all on wide range of species. Starting from 2017, I started to focus on only the insect genomics, because I started to work with a professor, work with professor at Florida and in Puerto Rico on several insect genomes. And then in 2022, I mean, I had to come back to India for personal reasons, and then I joined Atlas Skill Tech University for a shorter time before I joined Mahindra University. Now in the Mahindra University, I'm trying to set up a research program in insect genomics. So I have been working on different insects, primarily soybean aphid, cotton aphid, honeybees, springtails, thrips. But today I'm going to talk about mainly on honeybees and soybean aphid. So this is a tool that I recently developed, and it is now under review in BMC Bioinformatics. It is in the final stages of review. So basically, this is a tool to identify different types of honeybee species, honeybee populations. So using basically variation in at the genome level from different populations. So why it is important? So in US, the honeybees are important pollinators, are important pollinators, like for cashew nuts, almonds, and all these rich crops. So they need to have without. So what do? Is they carry in trucks these honeybee beehives and transport to the fields where the pollination they need that pollination. So for that one, so there are multiple types of honeybees, like European honeybees, Africanized honeybees, and Puerto Rico honeybees. So European honeybees, they are docile, gentle compared to Africanized bees. So the advantages of Africanized bees is they are resistant to different diseases, but European bees are not. So the Puerto Rico bee has both good traits of these two different types of bees. So there are many Africanized bees, but they are very difficult to manage. USDA goal is to identify geographic regions where these Africanized bees are prevalent, and then try to replace them with Puerto Rico or European honeybees. So, but they need to do screening of these regions. And also the border control, a tool is required to know what type of bee people are importing. So basically, we need a tool that can identify an unknown bee from its using the genetic information. So this is just illustrates how SNPs can be used to differentiate different types of bees. I'm sure most all of you know about it. Just a slide for those who are not from genomics background. So as you can see, the two SNPs can differentiate these different types of bees, like AT. If there is AT here, then we can say there's a German bees, GT like Brazilian bees, and A is Russian bees. So already there are millions of SNPs available in the public databases. For example, Wahlberg et al. This is a Nature paper published in 2014, where they identified 8 million SNPs in using 140 bees from 12 different populations. And Avalos et al. Is our collaborator, where they identified 2.8 million SNPs in 90 bees from 3 different populations. So there are already millions of SNPs, but that can differentiate these different populations. But main thing is we need a smaller number of SNPs that can be used to develop an assay than the people, and then scientists or the beekeepers can use to identify the different populations. So I I started with those two data sets, those 11 million and plus 2.8 million SNPs. Then my goal was to identify the smaller set of SNPs. So I performed this discriminant analysis of principal components. Basically, it is a combination of principal component analysis and a discriminant analysis. Basically, first it tries to identify few principal components that explains the majority of the variation. And then discriminant analysis is performed on those few principal components, so such that it maximizes the variation between the predefined groups and minimize the variation within the groups. So basically the variation within the, like for example, and this is a plot from the DAPC analysis, where here these are the group of samples from sub-Saharan Africa, and these are the sample genital African honeybees from Puerto Rico. So these analysis, once we begin with those 13 million SNPs, and then you need to identify those SNPs that can differentiate these groups, the top SNPs. So I had to run this multiple rounds to separate these different populations at different levels to identify final set of 272 SNPs. So these once I have those 272 SNPs, with the help of different cooperators, we collected samples from 20 different countries, total 874 samples. Yeah, samples from South America, North America, Africa, and Europe. And then we genotyped these. So we developed a panel using those 272 SNPs, a Fluidigm genotyping panel. Fluidigm is a platform where we can do the genotype of multiple samples. So we custom designed a panel using 272 SNPs. Then we genotyped this. Once I have these genotypes, then I did the clustering using those genotypes. Using these 272 SNPs, you can see these clusters. So they, for example, these two big clusters are like, one is the African cluster on this pink one, this blue one is European and related samples. And you can see that here even the Capensis cutillata, these are pure African bees. They are also, we can separate them. And America's Africanized bees, they are forming different clusters. Basically, these SNPs could differentiate the expected clusters. So what we could do using those millions SNPs, we could do using this smaller set of SNPs. Okay, now that we have these SNPs that can differentiate these populations, but we need a tool which utilizes this SNP data to identify an unknown bee. So I converted this cluster so that this is a hierarchical agglomerative clustering algorithm. So and so that cluster, using the information from those clusters, I generated this knowledge base network. So this knowledge base network, where the root node is all those 874 samples. And yeah, please note that these 874 samples are not part of those original samples from where I derived those SNPs, 13 million SNPs. So in the base, in the root node, I have these 874 samples with those 272 SNPs, genotypes for these SNPs. And then, yeah, based on that clusters information, I split them into these branches. So basically, when a new unknown bee comes, if you genotype that sample for these 272 SNPs, then so I wrote OR script to go through this network, checking basically which is the closest node this particular unknown sample is closely related to, based on the genotypes for those 272 SNPs. Then whichever is the closest, that will be the assignation for that new unknown sample. So as I mentioned, so this work is in the final stage of review in the BMC Bioinformatics. And I have also, the accuracy is 93%. How I calculated, basically, I took the known data sets, like the published data sets where we know what the bees are. And then I took the I selected, like in some data sets, there are only like 100 SNPs a part of my 272 SNPs. Even with those also, I could get a good accuracy. So based on the known information, I calculated like there are about 93%. So basically to improve the accuracy, which is what we are trying to work. Basically, we need genome-wide SNPs, more number of SNPs, or more populations, basically, more samples from these different populations. Then we can improve this knowledge-based network and improve the accuracy. That will be my future work. So that was about the honeybees diversity. Now I'm going to talk about the soybean aphids. So soybean aphids, these are serious pests in North America. I mean, not only in North America, but wherever the soybean plants are grown. So these were first detected in the US in late 1990s, or maybe in 2000. So we have samples collected from 2000 onwards, 2001 and 2002, and several time points. So we wanted to see how this soybean aphid has adapted to the new environment. And so the life cycle of soybean aphid is basically it has two hosts. In the winter, it goes to rhamnas, a buckthorn plant, and in the spring and summer, it comes back to soybean plant. On soybean, it grows in huge numbers, and it spreads. It invades the entire fields. Like in US, there are many hundreds of acres of land grown, so it spreads tremendously. So there was no genome assembly or any resources were available. So we sequenced and we generated next generation sequencing data. So Illumina paired end reads and Illumina mate pair reads with 3 kb and 10 kb insert size. And then that time we had 454 also, 454 mate pair data. And there was PacBio, that was the older chemistry of PacBio data. It was not of good quality, but still that was useful to get decent assembly. So we had like 3,224 scaffolds using this data. I mean, and recently we actually using the latest PacBio sequencing technology, we resequenced and we got full length chromosome level assemblies. Yeah, but this is what I have used to identify SNPs. So basically, apart from the genomic data, we also generated RNA-seq reads, and we from 3 biotypes, and then I identified 45,000 SNPs. Basically, the standard bioinformatics workflow for identifying SNPs by aligning the reads to the reference genome and checking the quality of the reads and checking the quality of the alignment and so on. And then we used this Illumina Golden Gate assay. We developed our custom assay using 3,000 SNPs, which had like best designable scores. And then we genotyped. So we collected samples from Australia, Canada, China, Indonesia. Basically, soybean aphid is not native to the US. It is basically native to Asia. So we collected samples from there, and we genotyped using the assay that we developed. And then after quality filtering, we are left with 2,380 SNPs from 3,791 samples. So our samples, mostly our samples, like as you can see, 2,647 are from US, because that is where most of our collaborators are. But still, we got decent number of samples from Asia, like 656. So we covered like China, Indonesia, Japan. All these are the regions where soybean senate has been growing, has been there for many years. So so for those SNPs, in those 2,380 SNPs, when I if for now you can ignore the why there are two colors here. So when I calculate allele frequency, so you can see that the ranges, it forms a nice bell-shaped curve with, so there is no problem here. But when you try to calculate those allele frequency in other populations, like other countries, like China, Japan, and all these countries, you see here, there are many SNPs with either 0 or 1 frequency. So that is odd, right? So why it is like that? Because we identified SNPs using the sequences from those biotypes from US and aligned them against the reference genome, which is also the American genome, American population. Then those, basically when we identify SNPs, like in silico, you identify those that are polymorphic. So when you take those SNPs and genotype them in other populations, they may not be polymorphic in other populations. So that is why we see this. You end up finding many of them with 0 or 1 frequencies. So that is ascertainment bias. So how do we minimize it or reduce it? I mean, to completely reduce ascertainment bias, you have to sequence all the individuals that you are working with. So which is not practically possible. So one way we could minimize is we selected. So here is a plot where is Asia and Australia. Basically, all these countries combined together, these are the allele frequencies. And then I chose only those SNPs with frequencies ranging from 0.3 to 0.7 and less than 0.8. Yeah. So if I so that those are the SNPs that are highlighted in green. If you see that the distribution of these SNPs, they are normally distributed in US, Canada also, and China also, Japan also, in all the samples, they are normally distributed. So these are the SNPs. There are 926 of them. So these are the SNPs with a minimum bias, ascertainment bias. So that is why we decided to work when we are, so for the downstream analysis, when we are trying to work on these Asian populations, we used those 926 SNPs. But for US, Canada populations, the full SNP set. Yeah, this. Yeah, the quality became poor because screen is big. So this is where then we try to, using those SNPs, try to understand the population structure. So here you can see, as I told earlier, we have a lot of samples from US, Canada. So these, so US samples, this violet color are from US, and this yellow samples are also from US, but these are the ones that were collected in 2000. That is the time period when when people first saw soybean aphid in the US. So if you see that the distribution of those samples in this PCA plot, these yellow samples are overlapping with the samples from Asia. So, but later on they are not, like those other than those from 2001. So that tells that these samples in 2001 are very close to those Asian samples. That means it is gives an indication that the origin of soybean aphid is from Asia. And here, if you look at these samples, so these are all these red ones are. You can ignore this box. That box was mainly from the paper. We wanted to highlight, enlarge that in the another image. Ignore this box. But if you look at the China samples are these red squares. So they overlap with all Asian samples except Japan. They also overlap with the US 2001 samples. So that what does that say? That basically these from China only, all the other Asian countries, the soybean aphid dispersed to other Asian countries from China, but not to Japan. Though. Japan looks like it has different population. When you look at this circle down here, these are the samples from Indonesia and Australia. So this suggests that Australia's probably from Indonesia, the samples were dispersed to Australia. So actually the results that we saw in the PCA plot, actually it matches from the FST values. FST values basically gives a quantitative description of the genetic variation between the populations. So when you see here, China, the smallest FST value. So in the value is low, that means the populations are very similar. If the value FST value is high, the populations are more different. So as you can see, China is the smallest between China and USA. Those are the samples with least FST value. So that indicates that China is probably the source of soybean aphid in North America. And the highest FST value is between US and Myanmar. So basically, if you remember in the previous PCA plot, so we have noticed that China is the source of all the other Asian countries. So Myanmar probably got different after the soybean aphid went to Myanmar. They probably differentiated further, and that is why they are so different. When we compare with all the Asian countries among them, like compared to China, so they are closest to China than to any other Asian country. So that again confirms what we saw from the PCA plot, that China is the source of soybean aphid for the other Asian countries. So based on that, we can generate a neighbor joining tree that tells the relationship between these different samples. As you can see, China is closer to all other samples. So this is, as I mentioned earlier, we have data at different time points. So we have from 2001 in the US, of course, even in the Asian countries also. So 2001 to 2013, we have samples collected. And if we, so this is a PCA plot. This is using only US Canada samples. So that is why in this analysis, I have used all the SNPs, 2380 rather than 926 SNPs. So in this, you can see those, there are 3 time periods 2001 to 2005, 2006 to 2009, 2010 to 2013. If you notice here, the third time period, those are less diffuse compared to the previous time periods. So I can like when you split this into 3 different ways, you can see here. I mean, these 3 are the same PCA plot. The first one showing highlighting only the first time period, and the second PCA plot I'm highlighting the second time period. The third PCA plot, third time period. As you can see, those third time periods samples are less diffused compared to the other. So this is probably like is a directive selection might be going on in the soybean population in North America. And this also reflects in this FST values. So the FST values are calculated from 2001 in US and to different years. Like as you can see, as the time period is advancing, the samples are more diverse compared to the original samples. So this pattern, we wanted to check whether the same pattern is observed in China and Japan, because we have data from China, Japan for these time periods. And we, as you can see, there is no such pattern that what we noticed in the previous plot. So basically, to adapt to the North American environment, so it is those population are undergoing a directive selection. So, I mean, we have also done several analyses. I thought I may not have that much time to discuss. We also looked at the genes, like the phylum reconstruction, at phylogenetic analysis. And we found that aphid proteome, there is duplication is there, and those are mainly those genes that related to apoptosis are present there. So that is probably an indication of possible adaptations to the plant chemical defenses and other forces that soybean aphid is facing. So the worldwide population analysis suggests that the source of North American invasive population is likely to be China or South Korea, and the genetic variation of North American soybean aphids decreased through time. Directional selection might have occurred. I would like to thank my collaborators, Dr. Rosanna Giordano, Dr. Tulul Jirai, and Dr. Jose Marcelino, and funding agencies for supporting the work. I'm happy to answer any questions.
Thank you, Dr. Ravi Kiran. I now request Dr. Padma Kumari, moderator of this session, to seek comments.
Thank you, sir.
Sorry, before that, I wanted to tell one thing. So the work that I showed is on soybean aphid or honeybees, but these can be used for any insect species. It is basically the fundamental is genomic data. So that can be applied. As I mentioned, I've been working with several different insects, but the underlying tools and the understanding is applicable. So basically, I'm looking forward for some interesting collaborations to work on some interesting insect genomics projects. Okay. Thank you.
Thank you, Dr. Ravi Kiran, for this interesting talk on genomes of insects and how they can be explored for their behavioral studies. Mahesh, can you go to the, Now the topic is open for discussion. Anyone can raise their hand or put the questions in the chat box, please. I have one question for myself from what is the basis for selection of the samples, insect samples? Because there are many stages in the insect. So Do you take all the stages, or what is the sample size at a particular location? How much sample do you need? Do you follow any protocol for that?
Actually, I'm not the one to tell that, because that is we have. I mean, I did not get involved in the sample collection part. I was mainly involved in the bioinformatics analysis. But what I know is, so we collected samples. I think at the same stage of life cycle, if that is what you're
They are probably what I understand is if they are prepared from those tip markers are from genomic libraries, then a stage should not matter.
Okay.
But if those libraries have been prepared from ESTs, or say this is primarily based on RNA-seq data, those differences could matter, because what we essentially analyze in RNA is not only the inherent variations, but also variations which arise out of post transcriptome splicing.
No, it's not RNA-seq data; it is genomic data.
Okay, then
but still, sir, they say male genome and female genome, there is a difference in size of the genome.
Yeah.
If that is the case, then the sex of the sample also would matter definitely.
Yes. No, this is at the same.
There is a difference in the genomes. Yeah, definitely it does matter for the.
Yeah, what I know is I don't remember what stage of the life cycle the samples were taken, but they're all the same stage of life cycle.
One important thing I'm able to draw from your studies, essentially, the patterns are more reflective of their divergence based on geographical distribution. But ultimately, like say, when we study populations, we should also study them in terms of their virulence or avirulence. Is there any efforts going on in case of soybean aphids to look it with another pair of glasses, the sequence data, whether their divergence is based on the kind of varieties or genotypes of soybean that is being cultivated in that particular locality?
I'm not aware of that. So, but the
you have one paper with different virulence, something like that.
Yeah, that is basically that is our collaborators paper. I worked on the analysis part. So yeah, there are there are multiple biotypes out there, and I mean I'm not aware of.
I know, like what I mean to say here is, you have like say very nicely using some of the most advanced tools, you have drawn some conclusions, but the same data, if it is looked at from the divergence of the populations in terms of evolution of a biotype, what I mean to say is at functional level, the study should be taken forward. That's my humble suggestion. For example, if we'll be doing this study anew, in case of say, we can certainly put for a joint R&D project or any other thing, maybe in rice, maybe in millets, or maybe in oil seeds. But our approach should be to look at the polymorphism in the genome, wherein functional diversity, signatures of functional diversity are there. For example, for bacterial blight, my colleague Dr. Laha, he is working with IRRI. They have a tool very similar to BID tool, what you have developed, called PathoTracker, wherein they are able to differentiate the populations. Say, for example, for management of bacterial blight, it is 100% through deployment of resistant genes. For example, in India, if, when they using a set of some 24 or 48, I don't remember it exactly, using a limited number of SNP markers, they are able to determine whether a particular population has already acquired the tendency to break down a known resistance gene, or they are trying to correlate that with the level of virulence of the pathogen. So I was just wondering that if we have to put up a combined project proposal in this regard, maybe it could be in any of the pest in rice, say for example DPH or this thing, wherein we should look at from functional divergence point of view also. That is what, and you have the set of tools, you have the knowledge, and we'll provide you the information about the compatibility or incompatibility of a population to a particular resistant variety.
Yeah, mainly, yeah, I need support in that. Basically, I don't know much about insect physiology and how the virulence and all those parts. I need, yeah, that is why I want to collaborate with people who have that.
Now the floor is open to entomologists, biotechnologists, and this thing. Sorry. Satyendra.
I have three questions. From first part of your presentation, you said that you could identify two SNPs to differentiate honeybee population. Those two SNPs are determining largely.
No, that was just an illustrative example. They are not real SNPs. Just to explain. Yeah. Those two SNPs is just for those with computer science background, like my colleague sitting there.
Okay, so you
to
could
make him understand, like
you could get close to 272 SNPs. So
yes, yeah.
So most of those SNPs are genic or non-genic.
That is the part I am going to work on now, like to understand what those SNPs are and the functional roles of those SNPs. That will be my
because that could have probably given more insight into understanding how those genes might be helping.
Because our main goal was to develop one tool to identify those populations. So now that would be the interesting. I forgot to mention that actually. That is the next thing I will be looking at.
And the second part of your presentation, where you were talking about soybean aphid, in the soybean aphid, you are mostly connecting SNPs with the origin of aphid, and connecting it with the diversity. But whether you have looked at the data from the point of view of the adaptation of these aphids in different geographical locations, whether some of those SNPs might be helping, and whether those SNPs are associated with some of the critical genes involved.
That aspect we didn't study. Those are all
because that will have more of practical utility.
Yes.
And then also if it has anything like Sir was also telling with the host and insect interaction, some of those SNPs determining critical. So anyway, you have not done. So the third question that I would like to know is, the insects are like evolving very fast. I would say after the microbes, probably insects are the organisms that evolve very quickly. And when you do such kind of studies, understanding and the dynamics of SNPs and all, how relevant it is, because over the maybe 1 year or 2 years, again, the whole population, there will be a lot of new diversity. So where it stands, your all this analysis and how dynamic these systems have to be.
Yeah, so that depends on the goal that we want to do. For example, if we want to understand how the current scenario and then, I mean, as you said, the insects evolve a lot. I mean, basically, what is the goal?
Could you identify in, okay, so coming back to that more precisely, could you identify in the genomes, in the whole genome, some of these regions which are more prone for this kind of changes, but some of the genome regions which are more or less conserved?
Yeah, that we can check those easily. Like for example,
like
we
the
have
insect is not even though selection pressure is there, but then insect is not ready to make the changes in those part of the genome.
Yes. Actually, I didn't talk about that work here. We are working on honeybee samples. We have honeybee samples collected before. In Puerto Rico, there was a hurricane Maria, which devastated the complete Puerto Rico. before Puerto Rico, before hurricane and after hurricane, we tried to study the genomic regions, which like where there was some signatures of selection going on. So we can definitely, if we have data from different multiple points or different conditions, we can check what is going on, which areas are more evolving and which areas are selected, and like that. Those things definitely can be done.
One last question.
Mhm.
It is generally in population genomics, particularly in human this thing, and also to significant extent in rice, rather than talking about single SNPs, we talk about polymorphisms with respect to group of SNPs, which form a haplotype. So any effort in this direction in bee genomics or bee population genomics to study the different haplotypes rather than looking at individual SNPs?
In the bee, we didn't do that because in bee, the longest, 500 bases is LD in the bees. I did this haplotype analysis in my PhD for cattle genomes, where in the QTL region, I developed a SNP map and then identified those haplotypes that are associated with milk production traits. So, but not in bees.
In rice also, for example, the LD value is as high as 200 or 250. Am I right, Dr. Kalyani? So the efforts are going on to develop the typical haplotypes based map, or rather SNPs. They are not talking about the SNP haplotypes rather than individual SNPs. I think considering that the insect genomes are smaller in terms of size, it will be more relevant for association studies. Say, for example, we want to differentiate one population from another or one subspecies from another. Rather than talking about individual SNPs, we should look at a group of SNPs which are co-associated in a particular species or subspecies or population.
Yeah, definitely. Yeah.
Thank you.
Students, any questions from students? Kalyani? Yeah, please.
You mentioned that in cotton aphids. Cotton aphids also you have mentioned. In your right line, cotton aphid also you have mentioned. So that data were not there.
That is
Still in progress.
work in progress.
Yeah.
So basically, we have the RNA-seq data, and that we have those cotton aphids fed on squash and cotton aphid fed on cotton. So we have those comparison. Try to understand how. So what are the differences in the gene expression? And when those cotton aphid fed on squash compared to cotton. So it's still going on. But no. Like Illinois.
So actually, I don't know whether I missed one part, but whatever you showed us population structure, that is basically PCA plots.
Yes.
There we are only clustering the group of samples collected from different regions. But the structure software where we actually basically with the plant germplasm and several material, with thousands of material we used to do with the same data, same SNP data or any genotypic data, we can cluster them and we will be able to find the admixtures also in the populations and how the gene traveled across or different like now what we are doing here, it is only the clustering based on the location. But maybe if you're, I don't know whether you presented that structure part. So structure part was there, means it will be more easy, and then functional associations and all will be easier, I think. So what we showed as population structure is actually PCA, principal component analysis clustering, like that.
Yes, structure is also.
Yes, structure software we are using is maybe in the different regions also we will be getting admixtures from different countries.
Yeah,
and
the main reason is we don't have that many SNPs for structure to work properly. We have like smaller number of slips. You may be working with larger number of slips.
Even with the
Even-
sets, we can actually try.
Yeah,
to
there are many programs structure and even there is so we didn't run. It can be done and yes.
What is the role of these symbionts? If you're taking whole genomes,
Mhm.
if you're clustering them, symbiont genome also would come into that, or you can filter it out. How do you?
Yeah,
Because
Symbi-
that's a different organ.
yes, correct. So so we when I assembled the soybean aphid genome. So when I did the assembly, so there are some programs called blob tools and all. They're based on the GC content. You can differentiate those rates that belong to symbionts, those rates that belong to the host genome. So at that level only is separated. Yes.
Then what about the methylation, demethylation? How significant is in these studies of genomes?
We don't have methylation data. Yes. So,
Do you get that methylation data?
That needs a different approach for SNP discovery.
yeah.
Okay.
It can be done, but it
Yeah.
requires a different approach.
Okay.
Yeah.
So students, any questions?
I can take the mic, sir.
You showed your proteomics study, based on proteomics study, you found some apoptosis related genes. Yeah. What is the trend of that genes? Means in apoptosis, so many genes are there in insects aspect, one will see. Then what is the expression dynamics compared to either China to other populations or from US to other populations?
No, we didn't look at the population level. We only saw at the genome level, like in the soybean aphid genome compared to other insect genomes.
Yeah,
Not
that's fine. Oh, yeah, any comparison, we showed you proteomic study. In proteomic study, you can see the dynamics.
No, we didn't do all those analysis. We just did phylogenome construction and saw that okay, there are compared to other insects, these have like more like duplication, and those mainly are apoptosis related genes. Like gene enrichment analysis, we did that and saw that apoptosis are more prevalent than other. So that's all. We didn't check in whether in China or other countries. What is the dynamics?
You found any inhibitor of apoptosis? Inhibitor of apoptosis related genes?
Inhibitor, that I don't remember. No, we did like we did not look at each individual. General apoptosis is a category. That
Okay,
we
only
found
function,
that,
apoptosis function. Yeah, if you see depth analysis, actually that is really very good.
Actually, we have a list in the table.
Yeah.
We have listed. Maybe their apoptosis inhibitor, or maybe I need to check.
Yeah,
I
you
can
mentioned
share
that when most China efforts are transferred to means
Yeah.
transferred to the means distributed other countries like US.
Yeah.
And then means it is you mentioned that adaptation, maybe detoxification from plant chemicals by detoxification function and these kind of things. You mentioned apoptosis. If you correlate these things, and then really it is very good information from scientific community. Then we can.
Yeah.
Now resistance mechanism is the big task for the scientific community. It's really very important thing, especially when you find some SNPs, either insecticide due to this SNPs, it is getting resistance, insect is getting resistance.
Yeah. Yeah, mainly I'm looking for my strength is bioinformatics. I don't know much about biology, so especially insect biology and physiology. So that is why I want to collaborate with you guys so that we can team up and make.
I just thought, are there any questions from online participants, particularly from other institutes like PJTSCU or IMR or IOR? Are there any questions or queries regarding the talk?
Then you can come. Any concluding remarks?
Dear colleagues who have joined us both online and offline, these are a series of seminars we have been conducting at least once in a month with varied subjects. And we always try to invite people with different sets of skill sets or knowledge base so that we get a wide array of information, and also we get diverse topics for discussion. I would like to thank Dr. Ravi Kiran for his very brief but very interesting presentation, highlighting the excellent work what he has done related to bee genomics and also related to aphid genomics. And I thank each one of you for participating in this thing. Now, Dr. Kalyani will offer a formal vote of thanks.
A very good evening to one and all. I, on behalf of our institute and the seminar committee, extend my gratitude to our distinguished speaker, Dr. Ravi Kiran Donthu, Assistant Professor, Computational Genomics, Center for Life Sciences, Mahindra University, Hyderabad. Thank you so much, sir, for sparing your valuable time for this important session on adaptive evolution in agriculture, pest and honeybee diversity, a genomic perspective. Your explanation was very fascinating and thought-provoking, and I think many of us would be provoked to undertake some kind of genomics in our insect biology, whatever we are doing here. I thank our beloved director, Dr. R.M. Sundaram, sir, for arranging this webinar. I also thank Dr. Chitra Shankar for nice introduction, the rapporteurs, Dr. Divya Balakrishnan and Shruti K. Dr. R.M. Kumar, Secretary SAR, for the webinar series which we are conducting under Azadi Ka Amrit Mahotsav along with our institute. I thank AKMU head, Dr. Shailaja and her team for arranging the online logistics for the webinar. On record, I sincerely thank all the participants, the scientists, ICAR partners, scholars, research scholars and students who gave an overwhelming response by attending in huge numbers. Last but not least, I thank chairperson of this committee, Dr. A.P. Padma Kumari Madam, for arranging the webinar on this useful and important topic. So thank you one and all, and we look forward for arranging one more webinar in our institute series. Thank you. Thank you.
All participants to be here with us for another 3 or 5 minutes. As it is customary, as a host, it's a privilege to do a small felicitation to Dr. Ravi. Anybody with photographs? I'd like to thank you for being with us for the whole of afternoon and then enlightening us. Thank you very much. Yeah. And as it's customary, we also give a certificate of commendation highlighting your talk and also kind of knowledge you distributed to us. Thank you very much.
Thank you very much.
I request online participants to switch on their videos for the last photograph. Thank you. Thank you, one and all. The meeting is closed. But it was wonderful. Now so many doubts.